山口大学共同獣医学部の豊福です。本日のテーマである環境検査、環境モニタリングという言葉は聞き慣れないかもしれませんが、日本では拭き取り検査という言葉で浸透し実施されています。ただアメリカのようにレディートゥイート食品を対象としてリステリアモノサイトジェネスに焦点を置いた環境検査、環境モニタリングはほとんど実施されていないのが現状です。レディートゥイート食品では加熱庫から包装までの工程で食品を汚染させないことが重要ですが、この工程はHACCPの CCP における管理手段では管理できません。一般衛生管理を確実に実施した上でHACCPに基づく衛生管理を行い、一般衛生管理とHACCPが確実に実施されていることを環境モニタリングで検証する必要があります。米国ではリステリアモノサイトジェネスが重要なハザードとしてハザード分析で特定された場合には洗浄消毒プログラムとして管理し、日常的に洗浄消毒後の機械器具の表面などを目視検査や ATP などでモニタリングし、さらに定期的にリステリアモノサイトジェネスをターゲットとして環境モニタリングで検証しています。リステリアモノサイトジェネスを対象とした管理ができていれば、低温増殖する腐敗細菌をコントロールすることもできますので、消費者への安全な食品の提供につながります。現状、リステリアモノサイトジェネスを中心とした環境モニタリングの実施は広がっていませんが、近年はさまざまなレディートゥイート食品が普及しており、フードロス削減の観点で賞味期限の延長も促進していることを考えると、レディートゥイート食品の中で特に冷蔵保管で賞味期限が長い食品はリステリアモノサイトジェネスの10度でも増殖できるという特徴を考えるとリスクが高いと考えられますので、重点的に管理していく必要があると考えられます。欧米への食品輸出をしている企業、そして要冷蔵で賞味期限が長いレディートゥイート食品を取り扱う業種業態、例えばコンビニ、スーパーおよびそれらの業態に納品する食品製造加工施設では、ハザード分析の結果、リステリアモノサイトジェネスが重要なハザードとして考えられた場合には、検証として環境検査を実施していく必要があります。食品工場にはリステリアモノサイトジェネスや腐敗細菌などが潜伏していることが多いので、まず自社の工場内を検査し、現状の近相データを踏まえて工場のハザード分析を行い、重要なハザードについては環境モニタリングを実践して洗浄消毒や二次汚染といった一般衛生管理でコントロールすべきハザードをコントロールし、それによって食品安全事故を未然に防げる管理体制を構築していくことが望ましいでしょう。本日の講師マーティン・ウィードマン先生を紹介させていただきます。ウィードマン先生はコーネル大学の食品安全分野の教授です。研究テーマは農場から食卓までの微生物による食品の品質と食品安全、および食中毒の原因となる腐敗菌や病原菌の伝播を研究するための分子ツールや定量的アプローチの応用です。彼のチームは350以上の査読付き出版物を発表しており、それらは1万回以上引用されており、世界中の微生物による食品の安全性と品質に関する課題の解決に貢献しています。最近では COVID-19 関連の問題に取り組む企業も支援しています。本日は環境モニタリングプログラムの重要性というテーマでお話しいただきます。
Hello everyone. So what I will cover in our first webinar today is the importance of environmental monitoring programs, which is really the setup to make sure that you can justify the importance of these programs, to acquire the necessary resources to implement these programs, these so important programs in different companies. So the take-home messages of today's webinar will be that food safety issues, both outbreaks but also recalls, are very often tracked back to failures of prerequisite programs and not failures of critical control points, which would be, for example, heat treatment. And these prerequisite programs very often relate back to the processing plant environment. And what I'm going to show you today is really the importance of the processing plant environment and other food-associated environments as sources of both pathogens, but also very important as sources of spoilage organisms. In order to address these issues, prerequisite programs need to be strengthened, and that would include sanitation, etc., and need to include monitoring, verification, and corrective action and corrections to deal with issues that arise from the environment. Based on this, environmental monitoring programs are essential verification tools, particularly in production of ready-to-eat foods that are exposed to the processing environment. And that has been recognized in a number of legislations and rules internationally, including the relatively new U.S. Food Safety Modernization Act. Based on what I will show you today, it will become very apparent, hopefully, that appropriate food safety systems and plans can provide significant economic benefits by reducing recall risks, extending product shelf life, improving food quality, and ultimately, very often, also streamlining production. And this is because food spoilage, quality, and food safety issues very often have the same root cause that lies in the environment of processing facilities. So here's an outline of what I will cover today. I will first talk about the changes in the environment around us that make environmental monitoring programs and food safety and food quality such important topics, and topics that are so much more important today than they were even 10 years ago. And then we're going to cover why the environmental sources are so important to address these issues and prevent food safety and quality challenges. One change that has occurred over the last approximately 20 years is really the development of improved tools to trace back contamination events with microbes in the food chain and to detect foodborne disease outbreaks, as is illustrated in this slide. What you see here from left to right, a number of different isolates of Listeria monocytogenes, a very, very important pathogen for the food industry. And you can see while all of these organisms are the same pathogen, have the same name, we can differentiate them in different fingerprint patterns, which just simply look like barcodes here. So you can see the isolate on top and the second isolate are different, and so on. And with this technique, and with these techniques, we can often differentiate Listeria monocytogenes in hundreds, if not more than 100, 400 or 500 different subtypes. Now, why are these tools so important? I'm going to illustrate that with the detection of a human disease outbreak, and this is really just an example of what happens continuously today, not just in the US, but in many parts of the world. So this is the situation we were exposed to more than almost 20 years ago, about exactly 20 years ago. And this is the number of listeriosis cases in the state of New York. Listeria is a rare disease, and what we can see in January of 97, it affected one person, the short bar, 2 people in February of 97, 2 people in March of 97. But in October of 98, we saw a higher number of human listeriosis cases than we'd seen the prior almost 2 years, 7 cases over that 1 month. This figure alone and this number alone really doesn't tell us what's going on. It could simply be a statistical blip, an increase that would go back down the next month, or it could be a bigger issue, an outbreak, where multiple people were infected from the same food that could become worse if it continues. With Listeria, it would be very difficult to differentiate this and identify this simply by classical epidemiological methods, where we would interview these patients and identify a common food they consumed. Why is this? Because listeriosis has an incubation period of ranging anywhere from 1 week to 2 months. Most people will not remember and cannot accurately recollect what they ate over 2 months. And more importantly, over 2 months, some foods will be consumed by almost everyone. Lettuce in many parts of the world would be a product that almost anyone will consume over a 2 months period. So if these 7 people all ate lettuce, it would not necessarily implicate lettuce as a source. But if we enter DNA fingerprinting, here's what we see. So these will be isolates with barcode fingerprint patterns, where on the top you might see an isolate from a human in June, another isolate from a human that showed disease symptoms in July, and you can see that these top 2 fingerprint patterns are different. But if you move to the bottom and look at the bottom 3 fingerprint patterns, these are 3 fingerprint patterns of Listeria monocytogenes isolated from 3 separate humans, you can see that these fingerprint patterns are actually identical. That suggests that these 3 humans were infected from the same source. Now, if we overlay this on our slide, we can see that this pattern, and you can see here that we had more cases, so 4 more patterns that have the same fingerprint. So in this case, we can see a large number of human Listeriosis isolates with the same fingerprint pattern. When we now overlay this on the graph of human disease cases, and we also draw it out further through February of 1999, we can see that 5 of the 7 cases in October were actually caused by the same fingerprint of Listeria, shown in this purplish color. The other 2 cases were caused by 2 different subtypes. What these subtype data shows us is that this outbreak or this issue actually started in September. We had 1 case with this fingerprint pattern in September, 5 more cases in November, 2 in December, 2 in January. So we can use these DNA fingerprint patterns to link together multiple cases that occurred throughout New York State. They were not in the same location, not in the same city, and they otherwise would have not been linked together. So based on this, we can identify that we have a considerable number of people infected with the same Listeria monocytogenes, most likely indicating the same source, which now we still have to go back and conduct epidemiological studies, but now we know the specific people we need to interview to identify a food source. So all people infected with the same fingerprint were interviewed to identify a common source food, and then further testing was conducted to collect food samples, test food samples, and ultimately find a food sample produced by a specific company that had a matching fingerprint of Listeria monocytogenes, indicating the specific plant that is responsible for this outbreak. Now, in this case, this outbreak did not just occur in New York State. It occurred in many states across the United States for a total of 108 human cases, 14 deaths, and 4 miscarriages, and it was linked specifically to meat frankfurters or hot dogs as a source of infection. With these tools, the outbreak was traced back to a specific plant in the state of Michigan. The important learning, another important learning, in addition to the fact that these fingerprinting methods allow for very effective traceback of disease outbreaks and identification of disease outbreaks, was found when the specific source plant was investigated. This plant was a plant that produced meat products. It had mandatory HACCP at this time, and it had an appropriate HACCP plan that was reviewed regularly by USDA with no concerns raised about the HACCP plan. But still, this plant caused 18 deaths linked to food produced in this plant, despite the fact that the critical control point, the heat treatment of the hot dogs, was under control, proper records were there, and the heat treatment effectively killed Listeria monocytogenes in the raw material. So what happened? How would a plant with an appropriate HACCP plan have problems like this? The investigation revealed this Listeria monocytogenes contaminated the product after the critical control point, after the heat treatment. The product was exposed to the plant environment. Listeria monocytogenes was present in the plant environment. It contaminated the product, not consistently, not all of it, but a certain percentage, enough to cause a large human disease outbreak over multiple months. And this is just one out of many examples and very similar stories where molecular subtyping methods, DNA fingerprinting methods, trace back outbreaks to processing plants where the critical control points were under control, but ultimately contamination occurred from the processing plant environment post-critical control point. These tools are not research lab tools. They're used in the US and internationally through a system that is called PulseNet, to a point where in many countries, virtually all isolates of pathogens, Listeria, Salmonella, E. coli, are routinely fingerprinted, and very often any isolates of these pathogens obtained from food samples are also fingerprinted and added to the same database. Now, over the last 2 to 3 years, we've seen a considerable change that is happening and occurring very, very quickly. And this change is due to the implementation and development of a new technology, which is genome sequencing. This is basically a tool that allows us to not just get fingerprint patterns of bacteria that are barcodes, but to determine every single A, C, T, and G in the bacterial genome, and to use that approach to fingerprint bacteria. This approach is driven by use of genome sequencing in human medicine. Today, a human genome can be sequenced for a cost of about $1,000. A human genome is about 1,000 times bigger than a bacterial genome. While costs don't fully scale like that, today the cost of sequencing a bacterial genome, depending on when, where, and how you do it, can be as low as $50 to $100 per genome and can be accomplished in less than 5 days. This is how the tool basically works. You determine DNA sequences for bacterial isolates that would be anywhere from 3 million to 5 to 6 million ACTs and Gs. You compare them, as you see in the bottom of this figure, and we can see that isolate 1 and 2 differ by one single nucleotide. It's an A versus a T, close to the middle of this comparison where the T is marked in red. And that will be one single nucleotide polymorphism. So it's a very objective way to compare bacteria and fingerprint bacteria and tell us same or different. This tool has been implemented routinely in the US since September of 2013, starting with Listeria monocytogenes. And what that means is every Listeria monocytogenes isolate from humans or foods in the US for more than 2 years has been characterized by whole genome sequencing. Same trend in Canada, England, Denmark, and actually a number of other countries around the world. What is the impact of these new tools? This shows the number of human listeriosis outbreaks from 1983 to around 2014. The yellow, fluorescent yellow bars indicate outbreaks. So what you can see is that before 1997, between 1983 and 1997, we saw 4 outbreaks, 83, 85, 89, 94. For an average of about 0.3 outbreaks a year. So 1 outbreak every 3 years. In 97 through 2013, saw much more outbreaks, except for 1 year, at least 1 outbreak a year, most years 2 or more outbreaks, for an average of somewhere 2.5 to 3 outbreaks per year. What happened in 97? Did our food supply in the US become less safe? Quite the opposite. What happened was we implemented better tools to detect foodborne disease outbreaks. And the specific tools implemented were pulse field gel electrophoresis, so barcode-based DNA fingerprinting. So we go from 1 every 3 years to 2 to 3 outbreaks a year. Starting in 2014, with implementation of whole genome sequencing, we moved close to around 8 outbreaks per year. So 1 outbreak a year before 97, 8 outbreaks a year since 2014 on average. That's 24 times more outbreaks detected in a given year. Now that alone doesn't tell the whole story. The blue line shows that the total number of listeriosis cases over the same time actually declined. So these are non-outbreak listeriosis cases. So we have less listeriosis cases, about 3 to 4 times less today than we had before 1997, but 24 times more outbreaks. What does that mean for industry? It means the risk of being detected as the source of a foodborne disease outbreak is at least 24 times higher today than it was pre-97. Probably more likely 80 to 100 times more higher if we account for more outbreaks detected despite the fact that there's significantly fewer human listeriosis cases. Bottom line is, food company today, unless their food safety system is 100 times better than it was pre-97, or at an increased risk of being identified as the source of an outbreak. How do we prevent this? How do we deal with this? we need environmental monitoring programs to detect Listeria and eliminate and control it before it contaminates the food. So the outside drivers of an improved public health system, not just in the US, but globally, enabled by new technologies, are a major, major driver that drives the interest and importance of environmental monitoring. Now, that is on the part of foodborne illnesses and food safety. A different driver, but equally important, forces and enforces the importance of environmental monitoring with regard to spoilage. And that's found in the importance of social media. This is an example of a Facebook shot that was posted by a Rachel Williams, who had a friend, Jennifer, who gave her son a Capri Sun beverage to drink. And the son basically mentioned that it tasted funny. When the mom tasted it, she said it tasted like alcohol. She put it in a glass, as you can see here. And you can see, if you look carefully, the cloudiness of the Capri Sun, which is most likely growth and contamination of yeast with subsequent growth of yeast. What happens when yeast contaminates a high-sugar beverage? It ferments the sugar and produces alcohol. So this does occasionally happen, probably always has happened in high-sugar beverages that are exposed to occasional contamination from the environment. But before social media, it was a single incident. Today, it is posted on social media. The specific post garnered at least 27,000 likes, 400,000 shares, led to a recall of Capri Sun product, led to a redesign, re-rollout with a clear bottom, new packaging that now allows consumers to look at the product first before they drink it, to detect potential spoilage issues before consumption. But huge cost for the producer of Capri Sun. And this is just one of many examples of social media triggered recalls and issues due to spoilage. Where do most of these spoilage organisms come from? Typically the environment of the processing facility. So two very different technologies, molecular biology, whole genome sequencing on the one hand, social media on the other hand, are driving the importance of environmental monitoring to prevent foodborne contamination of foods with pathogens and spoilage organisms. There was a time when occasional contamination with spoilage organisms was considered normal and not a large business issue. Today, it is a large business issue. One spoilage issue in one part of the country, another 1,000 miles away, would have never been linked together. Today they are, due to social media. And while some may argue that whole genome sequencing is mainly implemented in developed countries, social media are used throughout the world and will continue to drive this, and can easily also drive more emphasis on prevention of foodborne diseases, as these will also be reported through social media. So the environment in which we function as food processors today is very, very different than it is 20 years ago. And it requires new and different approaches to produce safe food, produce food that is unlikely to spoil or does not spoil, in order to prevent negative business consequences. Which brings me to the second part of this talk, is like, how do we prevent these negative business consequences? Where do these organisms come from? And gets me to the point, why environmental monitoring? Absolutely, raw product monitoring is important. Assuring your supply chain for your raw materials is important. But in many, many cases, the environment of the processing plant is the source of pathogens and spoilage organisms, as I will show you over the next few slides. This is some initial work we did with Listeria monocytogenes in a smoked seafood plant. You can see that we visited this plant over 3 visits, shown on this slide, 2 more are not shown. Every time we visited the plant, we collected samples from different environmental sources, but also from finished product. When we found Listeria monocytogenes, we fingerprinted it. Fingerprints are shown as barcodes on the right side. To make things easier when you look at these slides, we also, in the second column, put a name of the fingerprint, so you can see the first isolate is named 1039C, and put a star next to some fingerprints, actually next to all isolates that have fingerprint 1039C. What you can see is this fingerprint of Listeria was isolated in the first visit, again on the second visit, which was 5-6 weeks later, where we found it on finished product, product that is being processed, and the environment, and again on visit 3, which was another 5-6 weeks later, where we found the same fingerprint in floor drains and in various locations in the environment. Now, this fingerprint of Listeria was found on finished product where the raw material came from Norway, as well as finished product where the raw material came from Alaska or was wild-caught fish. So product from all over the world contaminated with the same fingerprint. What's the most likely source of this contamination? The processing plant environment, particularly since we also found that Listeria in the environment of this processing facility. In this specific case, we had further proof that the environment was the likely source. And that was because we sampled 3 plants within close proximity, about 20 kilometers of each other. The one I've shown you is plant D on the right column. Out of 229 samples, 10%, so 23 were positive for this one fingerprint. Plant B's and C, close proximity, less than 20 kilometers, we never found that fingerprint. It was unique to this processing plant. Plant C, you can see marked in red, 1046B, a different fingerprint, only found in this plant, found it on every visit, never found it in the other plants. Plant B has 2 Listeria that we find every time, 1042C, 1045. 1042C was found once in plant C, once in plant D. 1046B, 1045 was never found in plant C and twice found in plant D. All of these significant differences. So this shows us that different processing plants have very specific fingerprints of Listeria that live in these plants over time. And we sometimes name them analogous to pets, such as dogs and cats. Pet Listeria, pet pathogens that live in plants over time. So here I show you now a more well-designed and clearly outlined sampling plan of another processing plant. Every column is a month. On the top, you see sampling of the raw material. The next big block is sampling of collection of samples and testing of samples in the raw area. Next big block is the finished product area. Block on the bottom of food contact surfaces. And on the very bottom, we tested finished product samples, 6 samples per visit. What you can see first is, over these initial 12 months, we never found finished product positive for Listeria monocytogenes. We found Listeria monocytogenes in the environment, though, and every time we would find it, we would fingerprint it and mark different fingerprints of Listeria monocytogenes in different colors. So what you can quickly make out is that we have a number of different DNA fingerprints of Listeria monocytogenes, but one is most common, which is shown in a green color. That one is found not at every visit, but every other visit in the raw material area, but also found once in the finished product area on a door handle. Now, the major location in the raw material area where we found this is actually the apron of an employee. This is a single-use apron, so this Listeria is unlikely to survive on that apron, but somehow this employee apron picks up this Listeria monocytogenes from some sources, and it picks it up routinely. So we hypothesized that this Listeria survives in this plant. We collected more samples, particularly in the raw area, and of surfaces that were directly or indirectly in contact with this apron. This person picked up things from the floor, such as pallets, so we sampled the floor, we sampled other areas, and lo and behold, you can see that in one area, every time we collected a sample, we found it positive for Listeria monocytogenes with the same green fingerprint. What was this area? It was a floor mat. This floor mat is about 4 or 5 centimeters thick. It is foamy, difficult to clean. If it is sanitizer foam is put on it, it's unlikely to penetrate in the middle to kill Listeria monocytogenes. If the floor mat is submerged, the sanitizer is unlikely to penetrate the whole floor mat. But if a heavy person is put on that floor mat, Listeria that survives in the floor mat could come back out, recontaminate the environment. We removed floor mats from this processing facility. We worked with the facility to provide people shoes that were cushioned, provide the same cushioning as the soft mat. We never found this Listeria monocytogenes fingerprint in this plant over 6 months that we monitored afterwards, suggesting that these specific floor mats were actually the source, the hiding point of these Listeria monocytogenes. The importance of this is shown in the second 12 months that I'm bringing it back up here, where we see the same fingerprint now also found on the bottom in the actual food contact surfaces. So it migrated from the raw area to the food contact surfaces, showing that even an environmental contamination in the raw area can and likely will move its way around the plant to food contact surfaces, where it then will likely contaminate product. Note, though, that we've never, over 24 months and more than 120 food samples, found this specific Listeria subtype in the product. Does it mean it was never in the product? Unlikely. It just means it was at a low enough frequency that the finished product testing would not pick up this positive. The principle of the floor mat here is that there are locations that serve as so-called gross niches in processing plants. There are locations that harbor the organism, which could be Listeria or other pathogens, after the routine sanitation process has been completed, and therefore allowing these organisms to survive over time. The textbook example would be a hollow roller on a conveyor belt, as shown here. If bacteria penetrate in the roller cannot be effectively cleaned, but if this roller moves for 8 hours and rolls, any pathogens or bacteria inside may come out, contaminate the roller, contaminate the conveyor belt. If this conveyor belt carries exposed food, it will contaminate the food. If you take a hollow roller like this and you take it apart, you can see dirt inside, clearly indicating that the inside of this roller cannot and is not cleaned. So it's another example of a niche. There are hundreds, if not more, examples of areas in processing plants that can serve as niches that allow both pathogens but also spoilage organisms to survive over time. We need to find these, we need to identify these, and either manage them or eliminate them through environmental monitoring in order to prevent recalls and issues due to pathogens or spoilage organisms. That's why environmental sampling is so important. Here's another example to just show that this is not just recalls, not just research. This is an outbreak, 25 cases, 6 deaths in Finland. Most likely source of Listeria monocytogenes was survival of the organism in the screw conveyor in the butter wagon. This is notoriously difficult to clean equipment. While I've not been in this plant, don't have any first-hand experience, it is very conceivable that this type of equipment would be difficult to clean and would allow Listeria monocytogenes to survive over time, contaminate product over time, and cause an outbreak. This is not just Listeria monocytogenes either, as we will show you later. But here are some of the most extreme examples of what can happen with Listeria. This is one plant where the same Listeria survived for 12 years, 1988 to 2000. Same Listeria monocytogenes shown as fingerprint. This Listeria likely found a place in a plant, potentially things like floors, floor wall junctures, infrastructure like that is often linked to long-term persistence. Again, I don't know what happened in a specific plant, but this is actually not the only example of this. So these Listeria can survive for a very, very long time in processing plants if we don't have environmental monitoring programs that detect them and then take the corrective actions to eliminate and control these sources of persistent pathogens. Here's an example of salmonella, showing that this is not just a Listeria issue. 2 outbreaks, one in 1998, one in 2008. Same type of products, cereal, same processing plant, Salmonella agona, the serotype, same fingerprint both times. Pretty clear that this fingerprint of Salmonella survived for 10 years in this processing facility. Again, not unheard of, not unlikely to occur in other plants, dry plants, chocolate plants, powder plants, also pet food plants, examples of persistence of Salmonella there. So this is not just a Listeria issue, this is a broader pathogen issue. So I hope with this I've convinced you of the importance of environmental pathogen sources and environmental persistence of pathogens. Given you some examples of Listeria, I've also given you some examples of Salmonella, shown you that it's not just persistence, but that persistence leads to outbreaks and subsequent recalls and plant closures, or leads to recalls with severe financial consequences. If these outbreaks, if these sources in the environment are not detected early enough, and the only way to detect them, once again, is environmental monitoring. Now, we talked about spoilage issues. We talked about the Capri Sun issue, fungal spoilage, fungal sources, not just yeast, but mold spoilage, obviously common issue. Very often the sources of molds are the processing plant environment, without going into details. But there are also non-fungal spoilage issues that are very important in the industry. Here's an example. This is a dairy example where we were able to trace back a drop in shelf life, where milk usually had a shelf life of 17 days, had shelf life issues now with less than 10-day shelf life, to environmental contamination with a spoilage organism, in this case Pseudomonas. Pseudomonas survives very well in the environment, multiple examples of Pseudomonas as sources of spoilage. In this case, it survived in the rubber cup that was part of the filler assembly, and you can see a metal ring on the bottom, and we cut this open. There are small cracks there. Pseudomonas survived over time, contaminated the milk that was filled into those bottles with Pseudomonas, which grows at low temperatures, and then spoiled the product. We eliminated that source after testing. Shelf life went back to 17 days. So you can see where the squeezing action of this is likely going to open up some niches where Pseudomonas can survive, because it can get in there and can get out there if pressure is applied. But if it's submerged, this rubber cup is submerged in sanitizer, the sanitizer does not reach these locations because there's no pressure applied to it. Final example was one where we again found Pseudomonas. In this case, it was a Pseudomonas that produced color, blue color, contaminated cheese, not blue cheese, but mozzarella cheese, caused a discoloration that was easily detected by consumers, easily visually shared on Facebook and in social media. Major issue. Where did it come from? Processing plant environment. We used fingerprinting method to trace back the Pseudomonas sources to an overhead track that was used to move equipment and other things around, not regularly cleaned. The Pseudomonas survived there. With condensation, contaminated the product, caused color issues. We have seen virtually all colors one can imagine with Pseudomonas isolated from environmental sources. So a variety of different spoilage organisms, the environment is a very, very important source. The type of niches where these spoilage organisms survive are very similar to the ones where pathogens survive. So one method, pathogen monitoring, will identify potential issues with spoilage organisms, and the other way around. So therefore, these environmental monitoring programs will help you not just with food safety issues, but also with spoilage issues. We're back to the take-home messages. These are the same ones I showed you at the beginning. I hope this slide convinced you of the importance, or this presentation convinced you of the importance of environmental monitoring programs, and convinced you that they have broad benefits, not just for food safety. And in the beginning, I hopefully shown you and convinced you that food safety issues are very often tracked back to failures of the prerequisite programs, not your critical control points. Most estimates in the US right now are 95% of food safety issues come not from the CCP, but from other sources. We need to strengthen prerequisite programs, sanitation, sanitary equipment design, and then monitor and verify these, take appropriate corrective actions, but we need to have environmental monitoring programs to know where to focus. If we have these programs, we have appropriate food safety system and plans, we will get significant economic benefits, not just on the food safety side with reducing recalls, but also on the extension of product shelf life side, improving food quality side, and ultimately streamlining production. So as you can see, there are a number of very, very important reasons why environmental monitoring is so important and will provide economic benefits to industry if implemented and conducted properly. And that's, I hope, what I convinced you of this today. Thank you very much.
Thank you, Dr. Wieben. Now we'll start the Q&A session. For those of you in the audience, please submit questions through your browser window, and I will receive those. We'll start with the first question. Martin, for a food processor that has no current environmental monitoring program, can you provide some guidance to help them get started?
Yeah, so we actually run into this situation regularly where you have processing plants that do nothing or maybe do a little bit of ATP testing or very, very low level of testing. Typically, the first thing is just to very basic things. Assemble a team that is responsible for environmental monitoring that will see it through. Use a presentation like this today to really impress the importance of environmental monitoring to upper management. You need to have management buy-in in order to really implement these. Unless in those scenarios where your suppliers, where your customers will require environmental monitoring programs, which actually isn't that uncommon and becoming increasingly common that you have customer requirements for environmental monitoring programs. After that, it's really a question of identifying your major target organisms, so that depends on food, and then start with a monitoring program. Start small with a good plan. Plan out what actions you will take with positives. Environmental testing does not improve safety. It is what you do with the results. So you need to have clear action plans what to do when you find positives. And then get buy-in throughout the institution and try to also utilize issues that you see in your country with other plants that illustrate why environmental sampling programs are important.
Thanks, Martin. Next question from the audience. Can you discuss the new FDA Listeria guidance and your thoughts on it?
So the new FDA Listeria guidance, so broadly speaking, and so I want to try to make this relevant to beyond the US really. So different government agencies provide guidance on Listeria sampling and environmental monitoring. For the most recent one in the FDA was really a major change. So in the US, we had two different types of guidance over the last 8-10 years. One of them was by USDA, which strongly encouraged people to sample food contact surfaces. If a food contact surface was positive for Listeria species, USDA did not take this to mean that the food was likely contaminated. It just meant the processor had to take stringent actions and had to find the root cause and eliminate the root cause. But they did not have to recall product or did not have to necessarily determine whether the Listeria species was Listeria monocytogenes. The way FDA functioned, because they did not have an approved guidance out, and even today's guidance is really a draft guidance, is that if you found Listeria species on a product contact surface in an FDA plant, you were typically expected to test whether it was Listeria monocytogenes. If it was Listeria monocytogenes, you had to recall the product, which led to the situation where most FDA facilities did not sample food contact surfaces. Now, the new FDA guidance aligns very much FDA's guidance with USDA's guidance. Listeria positive on a product contact surface does not mean you need to speciate, does not mean an automatic recall. As long as you test for Listeria species, not the pathogen, you can Even have a second positive, you don't need to recall. It's outlined in some very, very nice flow charts. So anyone in the US or anyone who would fall under FSMA, I really strongly encourage you to read the FDA guidance. Again, it's a draft guidance document. But the point is it aligns FDA and USDA. We probably will see a lot more food contact surface testing in FDA plants, and FDA is now proposing that food contact surface testing is required, and it has to occur after 3 to 4 hours into processing. Typically, in the old days when FDA regulated plants did food contact surface testing, they tested maybe after sanitation. Now, it's very clear there's an expectation that your environmental monitoring program tests for Listeria monocytogenes, tests for Listeria species, tests during production, and test food contact surfaces. So it will change the picture quite considerably for FDA plants if this stays the same and it's approved. It's a draft guidance out for comments. I strongly believe it will stay the same. I hope it will stay the same.
Great, thanks, Martin. For the next question, for a company doing minimal testing today, how would you convince them to develop a comprehensive and robust environmental monitoring program? Or how would you convince them to invest? Or how can they convince their management in their company?
I think there's two ways of doing it. I think you can go the food safety route and the food spoilage route. You need to assemble case studies of the costs associated with both. This is much more difficult with food safety issues because a lot of plants say, well, we never had a problem. I think you need to assemble case studies from close related industries. You need to convince them that today is not the same what was 15, 20 years ago. I think you can take some of the slide sets that I use to try to convince them that the risk of being detected as a source of a disease outbreak is significantly higher, and the business risk of doing so is significant. Even if it's a recall without human cases, usually the costs are, at least for most, even medium-sized companies, $1 million. And most medium-sized companies will not survive these recalls and will not survive an outbreak. So it really is a question of essential, for business survival. On the other hand, you go a spoilage route. I think what we have seen with companies is if you have a good environmental monitoring program, your spoilage will reduce. That means you can extend your shelf life. You often will get better contracts because you have a product that has a longer shelf life. So their shelf life benefits from it. Again, you need to try to quantify them. You need to try to identify what their current shelf life issues are, and then convince them mechanistically that an environmental monitoring program will help with shelf life issues. And what that means is, if you have shelf life issues, what are the organisms, where do they come from? If they come from the environment, here's how an environmental monitoring program will help improve the environment and reduce the risk of spoilage. But in general, what we do every time there's an issue in a given country, in a given region that is linked to environmental sources, use it to train your customers and remind them that these sort of issues can be avoided with environmental monitoring.
Thanks, Martin. Another question. For a food processor that has already an established environmental monitoring program, how will this webinar series provide them with additional value or benefits?
So I think today's part of it is really to help them sort of convince management to continue to invest and potentially invest more into environmental monitoring programs. The rest of it, I think some of the key take-home messages will give them a better understanding of the different types of environmental monitoring people can do and what their real purpose and goal is. A lot of plants use different tests from anywhere from ATP to coliforms, Enterobacteriaceae, yeast and mold to pathogen, but they cannot always clearly map it, why they're doing the different tests, what's the purpose of the different tests. Another thing is that I think we've moved to a point where very often environmental monitoring programs are not well defined in their purpose. We can collect same samples or similar looking samples at the same location and test them for the same thing, but really trying to achieve different things. Sometimes we're trying to verify, sometimes we're trying to validate. This webinar will help people understand the difference between them and understand them and help them to assign clear goals to the environmental samples they collect and say, these are the actions, these are the goals, this is why we do it. And then reframe the environmental monitoring program as from, we do it to find problems, to we're doing this to verify, we're doing this to validate, and here's the consequences we take, and help them then align it also with regulatory requirements, such as FSMA, such as the new FDA guidance. Then the third thing really is at the end of it is, I think very often, the value of environmental monitoring program is not realized because the root cause analysis and the corrective actions don't happen. I think there's going to be a strong emphasis on that, and that will help companies truly realize the benefits of an environmental monitoring program, which often does not happen the way it should.
Thanks, Martin. So for our final question, what do you see as the next steps for our 3M food safety teams in the different countries to really take advantage of this trend of environmental monitoring?
I see 3M is in a unique position because you really have everything from sample handling devices to ATP, allergen swabs, to indicator tests, Enterobacteriaceae coliform total plate count, to pathogen tests. It is really a question of becoming experts and developing experts in the different regions and countries on environmental monitoring, working with in-country KOLs, working initially even with conferences to just educate people independently from 3M and but also with 3M on the importance of environmental sampling, and then becoming the experts that can help people with implementation of these tests, and being very aware of what's happening around, if there are recalls, if there are issues, and then showing people how environmental monitoring programs can help address those. So I think developing the in-house expertise in parallel of developing industry to see the importance, and then helping industry fulfill that need with the very, very unique portfolio we have. And then emphasizing really that it's a solution. It's not more money spent on testing. It's money spent on testing with a goal. And I think that's one of the big differences for me from finished product testing. Finished product testing detects a problem and doesn't point you towards solutions. Environmental monitoring, if done right, detects a problem but also immediately helps you identify where the solutions may lie and moves you much closer, much more quickly to a solution. And very often immediately tells you the solution. You have a problem in a certain area, it's not clean, it's pathogen positive, you can see it's uncleanable, you need to fix that. And I think that is something that's very, very important to emphasize.